So you have questions about mitosis and meiosis, correct? All right. So my guess is that what you have to remember is that when you're dealing with mitosis, as a review, you're copying the DNA. So you have one cell, and remember that mitosis is a division of the nucleus. And then there's cytokinesis. So there's the process there's something that's called the cell cycle, which is probably as important. And the cell cycle has phases, correct? And the cell cycle starts with you have G1, right? And here it's just growing. The cell is doing its job. And it's collecting, it's also growing. It's getting ready to divide. This can be they're varying amounts of time. There are sometimes cells go into what's called G0, and they pause the cell cycle. When they're pausing the cell cycle, cells like your neurons, these kids are so loud. Things like your neurons, all right, are in G0 for pretty much since you're born, all right, and they stay there. They do divisions, they divide, your brain grows, your brain develops, but at some point, the neurons stop dividing, and they don't start dividing anymore until you die. So that, again, remembering that makes sense because you don't want to lose your memory cells, the memories of your mother, your father. These are things you want to, and what's good to eat and what isn't. Those things you want to keep your whole life. So these cells kind of just don't do mitosis. They don't go through the cell cycle. So G1. The next phase after G1 is this S phase. Now S phase is where you duplicate the DNA, where you take DNA and you copy it. And if you watch the video that I posted, you remember that we use semi-conservative DNA replication, correct? Mm-hmm. So what happens is each of the new strands, when you do it, each of these strands gets a second copy made from it. So you read one strand and you make a copy of it, an alternate version of it. So you keep each of the old strands, and I shouldn't do it in this color. These kids are really distracting me. I don't know how you guys live a life that's not distracted. So each of the old strands gets a new strand made that matches it. So wherever there was an A, if this is A, T, G, C, right, this is still A, but then when it made the copy, it put a T over here. It's just it's the new version. Where the other one was T, what did it make what did it make? An A. But they're the same, aren't they? Right? They're all AT. It's just in the new strand one is used as a template to make the new strand. So this is semi-conservative DNA replication. And we talked about the experiment that led us to that, and I gave you a review video that discussed it. But in any case, that's all happening. During S phase, you're copying the DNA. Before you went into copying the DNA, you had two copies. One copy of a chromosome that came from mom. Let's just use red. I know it's a little sexist, but we'll go ahead and use mom like so. And just for graphic sake, and you have one from dad, right? One chromosome from mom, one chromosome from dad. During S phase, you copy those. So these are exact copies, but so this now is what the chromosome looks like after. So let me redraw it on the other side rather than do it that way. So before it looks like that. After S phase, you still have a copy from mom and a copy from dad. But now mom's and dad's chromosomes have been copied, and you have what are called sister chromatids. Sorry, I'm trying to diagram this. These are sister chromatids. Now the difference is that they both have all the same genes. If you find gene A here, you're going to find gene A on all 4 of them. A is going to be on all 4 of them. But the difference is they might be different alleles. All right, so, but now you have 4 copies of each gene, where before you only had 2 copies of each gene. You started with one, big A from mom and a little a from dad, and all the rest of the genes that are on that chromosome. And remembering with humans, it's 23 pairs, right, or 46. Over here, you still have 23 chromosomes, 23 pairs of chromosomes, one from mom, one from dad. But you have 2 copies of each. So really you have 90, so instead of 46, you have 92, right? 92 copies. But we still say you have 46 chromosomes because they're linked, they're connected together. You see that? You have this connection, this centromere that connects them together. So we consider them still 46 chromosomes. Give me a second. Ladies, please. So 92 copies, 46 chromosomes, okay? And they go right back to, hey. So now we take these 46 after S phase, the cell has to go through and start to, it checks. There's checkpoints here, right? So it's checking here. There's a checkpoint here, checkpoint 1, right? Then there's so it's checking to make sure it has everything it needs before it does anything. Then there's a second checkpoint here. And then there's a third checkpoint during mitosis, all right? So this over here is called, I guess I should do it all in what color? I don't know, some other color that's not this. So this is G0. This is G1. This is S phase. And now this is G2, right? And during G2, the cell's growing again, double checking, making sure it has copies of everything. But before it starts to make a division of the nucleus is so important, it checks one more time. So it checks one more time, and that time is one check in G1, then there's a check in G2, and then the last check is during M phase. So this is the third checkpoint. Sorry that it's so, all right, so there's 3 checkpoints in the cell cycle. And this is obviously the second to last phase, because ladies, please. So you have M phase, where, and again, here's a third checkpoint. You're checking during M phase to make sure that everything's still okay. An M phase is where you're doing your nuclear division, the division of the cell nucleus. All right. And then the last step of the cell cycle is, this is the last step, is cytokinesis. And that's where one cell finally turns into 2 cells. The nucleus here divided. So here, right before cytokinesis, you actually have a cell with 2 nuclei. And then it starts to pinch off in animal cells. It pinches off, and it finally divides in cytokinesis, in a process called cytokinesis, as the nucleus divides on both sides. So that's kind of the cell cycle. Those are the kind of big ideas. I hope that helped you. And then what's going on in mitosis versus meiosis? Now that's interesting. In mitosis, which is the process we just talked about in M phase. M phase the reason they call it M phase, it could be mitosis, it could also be meiosis. In mitosis, you go through the whole cell cycle and you end up with cytokinesis. And so you got one cell turning into two cells. And they're complete. And they're identical. And the cell cycle starts again, the whole cell cycle. So it could divide again, or maybe it goes into G0, but whatever it does after mitosis, these are identical cells. These nuclei are exactly the same, and so is every other component of that cell. They might differentiate, they might turn certain genes on and off, but their DNA, their nucleus, the code in their DNA is the same. So then, if we're looking at meiosis is, again, looking at it big picture, meiosis is really about sex, isn't it? And it's, in order to have sex, in order to have sex, you have to have two haploids. And that's half the DNA. So instead of 46 chromosomes, 23 pairs, you have to have you have to have a cell with only 23 single copies. So that you can combine that, if it's a sperm, with an egg that has, and let's show an egg the way it is, because eggs are much bigger, with 23. Oh, you're leaving? Please make sure you watch those videos and prepare for Friday, please. So that those two come together and they make one cell that has 46. Again, 23 from dad, right? Just like we talked about in mitosis a minute ago, and 23 from mom, the eggs and the sperm. The only difference between these 23, besides the alleles, right? This could have different alleles, which are different versions of the same genes. The only other difference is a big difference. Dad has an X and a Y, and mom has two X's. So dad is given this Y chromosome, which has different genes on it, to his boys. Otherwise, he gives to his girls that X chromosome. Does that make sense? Mm-hmm. So which one of these is dad going to give? Well, that's a question mark. It all depends on which one this sperm has. This sperm does not have an X and a Y. This sperm has an X or a Y, okay? And the mom is either going to give one X or it gives another X, either X1 or X2. I don't know which X it gives. It got one X from her mother and got the other X from her father. So her mom and dad gave her one X each. If this father gives a Y, this person becomes a male, theoretically, right? And if this person gives an X, then that person becomes a female. Genetically, normal development or average development. Let's say average development. That's how it's going to work. Okay? Is that reasonable? Yeah. Any questions up to this point? No. Okay. So mitosis, again, is making exact copies, but during prophase 1 of meiosis, you do something interesting. You do a recombination of the DNA. You take the mom and dad's chromosomes and you do something called crossing over. And that means that you take the two copies that you've copied in S phase, right? And they come together. Let me do the different colors, because I think it helps to see it in colors. Maybe it doesn't. I am wrong, but it seems to me like colors would help. Crossing over, if mom's two copies came together with dad's two copies, and they crossed over, wherever they crossed over, they're going to exchange this DNA. This is called a tetrad. A tetrad means 4. Right? So there's four sister chromatids. It's a complex with 4 copies of the DNA. Does that make sense? Mm-hmm. And they come together and form this tetrad. They do crossing over, and when they come out of crossing over, they split up as a group, as a pair. So then you have one pair that has some of dads and mostly moms on one chromosome, on one sister chromatid. And on the other, you have dads, what used to be dads, 100% dads, is now a little bit of moms and a little bit of dads, right? So now they're different, right? Where before these two were the same and these two were the same. Now all four are different. So during meiosis 1, because there's two phases to meiosis. Let's go back here and look at this. There's two phases. There's meiosis 1, and then there's meiosis 2, right? Does that make sense? Mm-hmm. All right. So in meiosis 1, what's going to separate is you separate the homologs. So here in mitosis, you separate the sister chromatids. They line up on the metaphase plate. You separate the sister chromatids. So you get all 46 copies during meiosis. You look at metaphase. So if this is the metaphase plate, right, the middle of the cell, all the chromosomes line up this way, with a sister chromosome facing each direction. All 23 pairs line up. All 46 chromosomes line up that way. I'm not going to draw 46 of them, if that makes sense. There's going to be 2 from mom, and 2 from dad for each one, right? And there's 2 pairs, there's 2 copies of each. They line up like that, and then the spindle fibers find the centrioles, and they line up, they attach to the centromere, and they get pulled apart, right? These things pull themselves apart. So in the end, what you end up with is a cell. On one side, you have a single copy, 2 single copies of one chromosome and 2 single copies. So you have 1 from dad and 1 from mom on each side of chromosome 1, 2, 3, 4, 5, 6, all the way to 23, right? 23 pairs. So you have, on this side, you have exactly the same, right? You have 23 on this side, exactly the same. Then you put the nucleus back around it and you separate the cells. And in the end, you get the same is exactly what you started with. Here, it's different. Here in meiosis 1, you don't separate the sister chromatids. You separate the homolog. So one of these is going to go in one direction, and the other is going to go in the other direction. They're going to go as a pair. So in the end, you have a cell, 2 cells, in the end of meiosis 1. On one, you'll have mostly dads and mostly moms and some of dads for one chromosome, some of dads. You still have 2 copies, right? Mm-hmm. But now, oh, that was a mistake. But now these 2 copies are different. And over there on the other side, I'm only using 1, but you have to imagine this is happening with all 23 pairs, right? And on the other side, you're going to end up with some of moms and mostly dads here and all of dads on the other one. So now, because they lined up, how did you do that? Because you started off, they lined up this way. They lined up as a tetrad. They lined up. Let me see if I can just do this on group. All right. Group and copy, paste. Good. And then move them over here. So they line up this way, you see. And because they line up this way, again, 1 from dad and 1 from mom, they line up like so, but they don't have to. Let me see if I can flip it. Yeah. So when they cross over, when they go over and they get separated, now the spindle fibers are attaching to 1 to each pair so that you get a separation that looks like this. Does that make sense? Yeah. So then you end up with 1 set of chromosomes here. Now you're going to do it again, but you're not going to copy the DNA. You're not going to go through S phase. You're not going to go through the cell cycle. See, over here in mitosis, they're going to go through the cell cycle. When they're done dividing the first time, they're done. Then they go through the rest of the cell cycle. They go through copying the DNA. They check all this stuff. Then they copy it. Then they split the nucleus again. Then they do cytokinesis, and they're ready. And they just keep multiplying the cells. So these are all exact copies. Here, after meiosis 1, you have 2 cells. They're not identical. So and let me put that in an asterisk. Let me or zoom in. I guess that's better. 2 cells. They're not identical. Let me put it on the outside. I guess that makes more sense. Not identical. And they have 23 chromosomes, technically, right? But 46 copies, because these chromosomes are still linked. They're still doubled up, right? Because they're still connected. And here you have 23. They're not identical. They're mixed. The DNA has been recombined. You have increased genetic variability. You have changed up the DNA. That's why the children don't look exactly like their parents. They're never like, you never see like half your mom and half your dad in one person. Because any trait, you could have a mixture of the two, depending on which allele you got with which chromosome. So here you have this DNA. Let me go ahead and just copy it again from over here. And sorry, you go ahead and erase. Oh, I didn't want to do that. And let me erase this one here. And then erase this one here. So we got this one. So you got this one here and this one here. Let's zoom in. So over here, you're going to have 23 that maybe they look like this, and this one over here looks like this. And so you're going to have different combination of alleles. Does that make sense? Mm-hmm. So you have a different, I can't believe it. Why is this so hard? I don't know. So you have one here and one, oh, why is it not, it's so, what? Well, all right, cooperate machine. All right, let me try this. Sorry, give me a second. I'm trying to make this group up. All right, so here where they had, in mitosis, they would have lined up, you would have separated the sister, you would have 23 pairs. Here you still have 23 pairs, but they're connected. So you might call them 23 chromosomes, depending how you look at it. But there's still 46 copies. It's just connected. So there's 23 chromosomes, where in mitosis you'd be done. They would be separate chromatids, separate chromosomes, one copy in each chromosome. Here you have 23 chromosomes, but they're 2 copies of each. And they're different. So now you're going to go ahead and do everything like you did in mitosis. They're going to line them up on the mitotic plate. Now the sister chromatids are going to get separated. So now you have, at the end of meiosis 2, let me move this down here. Each of these is going to divide again. At the end of meiosis 2, each of these is going to divide again. Oops, there should be 4, right? At the end of meiosis 2, each of these is going to divide again. But in each one is going to be a different outcome. This one's going to form this. And in this one, you're going to have all 100% that. And then the other one, you're going to have 100% that. And the last one, you're going to have this. So now you have 4 different sperm or 4 different eggs. That's just with one chromosome. And you can imagine that happening with 23. So there's 20, in each of these, you're going to have 23. So half the normal DNA, half the autosomal DNA, 23 in each one, correct? But even though you have 23 in each one is also different. So you can imagine 4 sperm, 4 different eggs every time there's sex. So that's why that's why sex is such an advantage, because it allows for differentiation. It allows for a much bigger difference when you get these 4 possibilities with 4 possibilities from the husband or wife, and they make a baby. That's why every individual is different, because you're mixing up the DNA so radically, let alone mutations, which could also happen. So when you're looking at complete, in mitosis, they're completely identical. In meiosis, there's 4 cells at the end of meiosis 2. They're all different. In meiosis, you have crossing over, which is what makes them different. And also there's independent assortment. So which one of these goes into each egg or each sperm? But then you have chromosome 2. In this one, you have mostly moms. In this one, you have 100% moms. Here's 100% dads. But the next chromosome might be opposite. Maybe all of dads goes in chromosome 2 here, and all of moms goes in chromosome 2 here. So it multiplies the variation. Where in mitosis, they're exactly the same. Okay? That's mitosis and meiosis in a nutshell. And remember that when you're doing Punnett squares, what you're doing is you're looking at the possible combination of these alleles. So when you're looking at, let's look at a dihybrid cross, which is why I like to do the dihybrid cross in a lengthier way. When you're doing, maybe you have big A, big B, little b, you're heterozygote, and you marry someone who's heterozygote, and you both decide to have babies with each other. Well, which egg, which sperm, which haploid cell are you given to which to make the baby? Well, it all depends, because you could have big A, big B in one, and the other one you could have big A, little b, and another one you could have little a, big B, and the fourth you have little a, little b. The same with the other person you're marrying. Okay, and so at that point, the combinations can be multiplied. So when you do a Punnett square, that's why we do a dihybrid cross, where you're like, well, I could give my child this, or maybe I can give my child this, or maybe I can give my child this, or maybe I give my child this. And again, these are just 2 genes, right? You imagine 10,000 genes. And why that? I hope you understand that that's why no 2 people are alike, because it's 10,000 genes and you're recombining each set. So then your husband or wife could do the same thing, and then you could have 4 different possibilities, right? You can have big A, big B, big B. We've done this before. I don't want to go over it again. We've done this recently. You understand this, right? This dihybrid cross concept. So then you can have up to, 16 different individuals based on this. Just these 2 genes. To multiply that times 10,000, it's that you can have as many people as there are probably stars in our sky, right? So over time. So that's it. That's mitosis and meiosis in a nutshell. Did that help you?
Yeah, it did.
All right, excellent. I'm going to post this, okay? Mm-hmm. All right, thanks.